As he said, I'm Elisia Clark, and the title of my talk is Research Resources for Tool Validation through the Michael J. Fox Foundation for Parkinson's Research. So I'll just briefly start with our mission and vision. We're dedicated to finding a cure for Parkinson's disease through an aggressively funded research agenda to ensure the development of improved therapy for those living with Parkinson's today. We are a public charity that was founded in 2000 by Michael J. Fox. More than $1.5 billion have been funded to date. And in 2023, we funded over $225 million in research, spanning more than 450 grants. And each year, that number continues to grow thanks to our donors. 33% of our projects are funded to grantees outside of the US, but over the landscape of what we fund, it ranges from academics to biotechs to pharma. And then our strategic research vision is centered on advances in disease definition, measurement, and treatment. And so with that, we envision better PD treatments, better PD trials, and better PD healthcare. And so a lot of our programs and initiatives are centered around our guiding principles, which is people-centered research, leveraging resources, diversity, equity, and inclusion, external validation, open reproducible science, and impactful partnerships. And so when you think about the therapeutic pipeline, we like to think that we're moving our projects through this pipeline ultimately to de-risk the PD field for the larger funders. And so we do this by addressing our field-wide challenges, aligning the field around priorities, investing in the riskier projects, and providing non-dilutive funding. So today I want to describe for you what our preclinical tools program is and how we support tool validation through these programs. And the program started because of a number of problems that were identified both at MJFF and through the concerns of researchers. And it was around the lack of tools, the lack of QC and standardization of the tools, access issues, and slow research progress. And so these are some of the approaches that we came up with to try to help solve these problems, which I'll go into a little bit more detail in the next few slides. And so our program has 3 pillars or 3 arms, and below here are the different tools that we support, and we're always looking to expand as they become relevant to studying Parkinson's disease. So I'll first share with you our new program, which is called the Tool Evaluation Program. This started last year in 2024. And the goal of this program is to fund researchers to do head-to-head comparisons of commercially available tools. And the goal is to produce a resource that can be available to inform tool selection for researchers' specific tool needs. So currently we have a project that is comparing the synuclein triplication iPSC lines from the various commercials companies. We have projects going on for different rat models, and then we also are partnering with Icarus to do antibody head-to-head comparisons for a select number of targets for one of our other initiatives at the foundation. The next one is the Sponsored Tools Program. So we recognize that in order to share resources and have open science, it takes time for labs to fulfill these MTA requests. And so our goal is to kind of protect that time for researchers to spend on their hypothesis-driven work. And so we will help fund the transfer of any tools that were generated in academic labs to repositories so that they can handle the sharing and all of the streamlined distribution. And then finally is our tool generation program. And so in this program, we actually fund and partner with CROs, the generation and development of tools. And a fundamental key point of this program is that once the tool has been generated, we partner with academic collaborators who will then go on to characterize those tools. And so it's not until that work is done that the tools are then given the green light to be distributed at these CROs. So within our program, we have a catalog on our website that lists all of our tools that either we funded or we have supported. More than 50 tools are in development right now. 200 tools have been made available, and currently more than 100,000 units have been distributed. And so I just want to talk a little bit about our antibody program and what this looks like in real time. So in our first step for tool antibody generation, we've generated over 45 novel antibodies with different partner companies, and we have a few in development right now. As I mentioned, that next step, it then goes on to be externally validated with academic collaborators, testing in various complex systems with different partner labs. And then once that is done, those antibodies have then gone on to be distributed at 4 different partner companies. And so this work has been done with various collaborators and partners, plus many more that are on the screen that we say thank you. And obviously the goal is to have high quality reagents to speed research, enable those new discoveries, and improve replication across the labs within the Parkinson's disease research community. And so where we see ourselves fitting within the research ecosystem is that we like to be the ones identifying these key gaps in the field, as well as hearing from the community of what these gaps are. We then partner with academic and industry groups to fill those gaps through knockout validated testing, with the goal of creating that publication and community access, and then providing article processing fees, because as we know, that could be a barrier sometimes to the sharing as well. And so finally, just want to briefly go over some of the challenges that we see at the foundation around tool validation and replication, and ways in which we have tried to find solutions. So the first being, I would say, the lack of funding and incentives for researchers to actually take time and perform these replication and validation studies. And so at the Fox Foundation, we created this initiative where we actually provided grants to to grantees to perform these replication studies of the high impact publications. Also, there are journal publishing preferences where we know they tend to want to publish more of the novel studies, not so much the replication studies. And so one example is when the foundation partnered with the Journal for Parkinson's Disease to actually pre-approve the publication of our funded replication studies. And this was actually received so well that this particular journal in 2023 created their own program, and they actually created replication studies as a category for their journal for people to submit articles for. As we all know, there are so many tools to choose from, and often researchers will come to us asking, which antibody should I use? Which model should I use? And so our hope is that our tool evaluation program will provide that resource to inform tool selection for researchers. And then finally, unaligned protocols. We'll often see, even for the same tool, people using different protocols. And so when we can, we try to work with researchers in the field to try to come up with resources that we can post on our website to kind of align on best practices or standardization around these topics. So finally, my last 1 minute, I just wanted to point out that as a funding agency, we do see benefits in funding research tool validation. It provides that trust and reliability to the research community. It sets the high quality research standards. And then there's obviously that open science component that we value and we know the community values also. And so finally, I'll just leave you with a few more resources that you can find on our website. There are links to our different funding callouts, our catalog to all of our preclinical tools and models. If you want access to patient biosamples, data sets, or clinical trial resources, they are there as well. And so with that, I'll say thank you.